Okay, it's 11 a.m. Good morning, good afternoon, or good evening, everyone. My name is Cherry Kubota. I'm a host of this Indoor Ag Science Cafe series. We are doing this monthly basis to share science-based information and then also most updated technology status. Funded by USDA grants. So the three groups are participating to do this, ADVANCEA group, OptimIA group, and then also CEA HERB. We have multiple speakers from, already this year from academic community of CEA, and then also we accept speakers from industry. So if you're interested in participating, please let us know. The idea of this webinar series is again sharing information and then basically assisting each other to move the industry forward. So just wanted to give you the upcoming webinar series. So today, as May 6th, we're going to have Angela Walla from Texas Tech University and then her graduate student, Grace Akumu, talking about food safety research update for hydroponics. And then also June, we have another webinar. It's about 1 month from now, June 10th, introducing company Canopy Technology from David Ashton from Canopy. And then July, we don't have any cafe. This is kind of our tradition. We started many years ago, 2000, I don't remember, 16? Um, or sometime around that. And yeah, so we have a tradition of not doing any webinar in July. So July is a conference month and everyone is busy. So and then August, we have Dr. Kelly Walters. This is also supported by CEA Herbs Group. She's going to talk about germination techniques for herb seeds. So it's going to be interesting. So with that, I'd like to have Dr. Angela Walla and Grace Akumu from Texas Tech University talking about food safety research recap. How do foodborne pathogens survive in NFT and DWC systems? So NFT is a nutrient film technique. DWC is the deep water culture. Those are the typical or widely used systems for especially leafy green hydroponics. So with that, I like to have Dr. Walla. I'm going to stop share, and then she and Dr. Walla and Grace take over.
Well, good afternoon, everybody. I'm hoping everybody is having a great day. We're going to talk a little bit about food safety. We're going to focus on two different topics of NFT or nutrient film technique, and then our deep water culture system. So our journey today. Number one, we're going to talk about kind of the history or where we are within hydroponics and food safety, some of the patterns that we've seen, and then we're going to jump into some of the whys. Why are we seeing these things? And then the power of a risk assessment. And then we're going to dive into some of the most recent research. I am here with my PhD student or PhD candidate, Grace Akumu, who will be joining me, and she'll be discussing a lot of her research later on in the presentation. So please use the chat feature and ask any additional questions, and we'll answer those at the end. So let's start here. Well, number one, let me introduce myself. I'm Dr. Angela Walla. I'm a professor at Texas Tech University, and I've been working in the CEA space, and I was trying to calculate in my mind since about 2005 is when I started my first project working with hydroponics. Before that, I worked a lot with outdoor growing and produce. And prior to that, I was within the meat industry, helping them on the food safety side of things. So around 2005, 2006, we started to see an increase in foodborne outbreaks related to produce. Everybody has probably seen this slide a million times of which produce items have caused the most outbreaks. That mighty result that came out in 2009, that about 46% of all foodborne illnesses came from fruit and vegetables. And then 2021 is when we saw that the mighty outbreak with salmonella, leafy greens, Bright Farms, which we were really surprised. They have an excellent food safety team and excellent controls in place. And all eyes went on to them to see what was the root cause for that outbreak. There was no conclusions really, but they found salmonella in the pond, growth media, preparation area, water management, and sanitation. If you're in this space, probably not really surprising of seeing kind of where it would come. We're going to talk about that today. Where does it come? How does it survive? Now, this comes off of earlier outbreaks, and so I want to bring in some outbreaks that maybe you've seen or seen the results of. But in April of 2013, I remember reading about cucumbers and greenhouses and this outbreak that occurred there. And then in 2014, there was another cucumber salmonella outbreak with the same strains that came in 2013. And then later, there was another cucumber outbreak in 2015, similar. 2016, we saw some more, 2018, and now in 2024, we just saw a cucumber outbreak that came out. The reason why I bring this up is it's important to make sure that you're watching the patterns of what products are being affected, what pathogens are being affected. Is there any commonality between them? Because now onions have gotten into the works, but we've had a lot of onion outbreaks over the years. From 1998 to 2013, there have been over 7,000 people that have been infected with onions. And so I'm seeing an increase in the number of controlled environmental ag now going towards onions. So is it a hazard? Is it not? Something to consider as we look forward to food safety concerns and products. Now, tomatoes, something else that is highly grown, we see within the CEA. I'm going to use CEA, Controlled Environmental Ag Systems, but the outbreaks have been going up and down over the years. And so when we look at these patterns and now we're at a low, I expect to see some outbreaks coming up soon because of just the patterns of how they survive in the environment. Similar with leafy greens, we had a big null of no outbreaks. And then in 2006, 2007, we saw it started to spike up with the number of leafy greens. And how many Thanksgivings did we go through where there was no romaine lettuce on the market because that was the time that we had seen the outbreaks? Most recently, and the reason why I'm here is because I'm on the CEA herb project, we've seen a lot of outbreaks now with our herbs. Again, with all these products, what is the common thread amongst them? They're raw, they're not cooked. A lot of times they're garnishments going onto the top. But this investigation that the FDA did that was published finally in April of 2024 revealed that Salmonella, E. coli, Shiga toxins, Cyclospora are concerns when we think about basil, parsley, and cilantro, and some of these fresh herbs. So that's both domestically grown as well as imported into the country. So this is my chart. So when people ask me, what are the pathogens I should be concerned about if I'm growing hydroponically within that? We know Salmonella has a huge history. We've seen some with E. coli and Stex. We've seen some evidence of Listeria monocytogenes, but when we think about the major pathogens, norovirus, cyclospora, hepatitis A, are those ones that we should be concerned of as well. So why? This is the question I get asked so many times in daily, but here we go. When we look at plants and what they need to grow within a hydroponic system. In general, they need macronutrients, they need micronutrients, beneficial. But also when we look at our foodborne pathogens and bacteria specifically, so we're looking at E. coli, Salmonella, Listeria, they need a lot of the similar ones for them to grow and survive. So when we do an overlay of what are the nutrient needs, other needs for a plant to grow within the system and for pathogens to grow, I highlight them in yellow. Look at all the overlap. So as you're feeding your system with all of those rich nutrients, as well as you're putting in the sunlight and the temperature, you're controlling the pH, you're also making it ideal conditions for the foodborne pathogens that, if introduced, and we'll talk about the inputs, if introduced, are able to survive in that environment. So the nitrogen, the phosphorus, all of those are things that the pathogens love as well. When we think about E. coli 0157H7, I'm just going to give those that may not be as food safety or microbiology sound. E. coli has a great ability to recover. So if you were to put this foodborne pathogen in a harsh environment, it has mechanisms to have DNA repair, to stabilize itself if it's at a high temperature, and it has the ability to go dormant if you put it in unfit conditions. One other amazing attribute, and I'm going to say amazing attribute of E. coli 0157 is it has this ability to shift from a viable but not culturable state to a viable state where you're able to detect it within the system. Similarly, when we talk about salmonella, salmonella has an amazing ability to form biofilms. It has a way of basically taking over a system, plant system, and really shift the host to allowing it to survive. Like E. coli, it is also able to go to this viable but not culturable state if it's being has high temperatures or low water availability or high salt availability or some of these extreme conditions. With Listeria, Listeria is the queen of biofilm formations. It's the queen of also going into not requiring oxygen state, and it likes a large ranges of temperature, which is why Listeria is usually associated with air conditioning units, your refrigerator. So in the drain, it can tolerate those high salt, high pressure, heavy metal conditions, and it's really tolerant to UV light. Which is why a risk assessment must be done by companies to determine the severity and the prevalence of all of your inputs going into the system. So when I work with a lot of controlled environmental ag companies, especially in the leafy green space, they always rely on their certification of analysis to certify that everything coming into their establishment has met specific standards. But through this presentation, I hope you will look more closely at those CEAs that you COAs that you have, and look at what specifically are you requiring, because we have a lot of inputs that come into the system. We have the seeds, we have fertilizers, the herbicides that you might be utilizing. You have the whole nutrient solution and all those nutrients being put in. We have water or agricultural water. We have substrates. We have the actual growing unit and all your holding tanks. We have tons of surface. So whether that's a table, whether that's tubing within there, we have equipment, we have the actual facility itself, those walls, those high ceilings, that piping that might be up there. And then we have the management. We have the people, the sanitation, your buying criteria that go in, as well as your maintenance. So when you're doing a risk assessment, each of these have to be considered. And we know that through research, which you're going to learn more about today, is not every unit is the same and you are different. You have different inputs, different systems, different ways of growing your produce that have to be considered when you are doing your risk assessment. Today, we're going to focus on the nutrient film technique and deep water culture with our research. So I'm going to give a caveat as we dive into this research is that some of this research is currently under peer review. So we ask people to respect that factor. And we will share out all of our citations once that has gone through the peer review. But we wanted to provide some highlights. So let's start with water. And this is some of my earlier work. This is going back to 2016. And we really wanted to find out what happens if you just put E. coli, if you put salmonella, if you put in non-0157 E. coli into a water system, what happens? And let me orient you to this graph. On the x-axis, we have our time in hours, and on our y-axis, we have the microbial counts. The red lines are the control. So that is just pathogens in the system. We're just monitoring the water as such. When we add in the nutrients into the system, just doing a nutrient solution, running it through the system, that's what you're going to see in your black. And then the purple is we were growing lettuce in the system, and we're looking at the interaction. What you can see by the chart is nothing surprising, that the pathogens grow over time. When you add in the plant into the system, there's that competition with the additional bacteria, and we see a little bit of decrease, but for the most part, the bacteria is growing over time. Similar state, almost the same graphic, we see that with salmonella and how it grows in the system. So some of our major takeaways from our initial work is that Salmonella, E. coli stacks, as well as Listeria, I didn't show that work, but they survive with or without nutrients for at least a week in the system. They survive longer in just the nutrient solution. So when you're thinking about your holding tanks in the unit, they love that, especially if you have those air stones in there. They love to survive by there. When you add in the plant system, there's more competition, but we've actually found that they're able to survive over a month within those systems. So let's talk about the seeds. So let's start at the very basics area of as you're growing. So we talked about water, let's go over to the seeds. This is some of the work that we've done with herbs. And so we wanted to look at that germination phase, the first 14 days as you're germinating, you're getting to your 3 to 4 true leaf state before you are implanting into the system to see just how E. coli and salmonella will survive. We've done some of the similar work with lettuce, and we see the same behaviors in just the seeds and survival. So starting at time 0, we started at a higher inoculum level at about 6. By the time we were done with day 7, we had about 1 or 2 true leaves at that point. It started to be about steady in the system. And we know that with E. coli, it has an amazing ability, especially with this in the seeds from previous work, to be able to internalize in during that time period and thus be able to sprout out later. When we look at salmonella, we found the similar pattern. We started at a lower concentration to see if the lower concentration would affect the survival of salmonella in this case, and we didn't see any difference. Once salmonella is introduced into the system, it is able to survive all the way through day 14 of the system and the seeds. Similarly with basil seeds, they love to swell when we have water presence, and we see also that internalization of salmonella being able to go into the actual seed during that time period. What that work also showed us was that once it's been internalized, when you do additional washes on the outside of that seed afterwards, it's not easy to kill the actual E. coli and salmonella that is inside the actual seed. And so you'll see it sprouting out and coming out in the actual plant later. We did a little bit of substrate work early on, mostly with microgreens. As you know, within the CEA space, there's a lot of different substrates that you may be using or growing media that all have different water holding capacities, pHs, drainage abilities within them. And not all substrates are the same. And this is, and I see my colleague on the line here from University of Arkansas. Shout out to Kristen Gibson and all the work she did with substrates. Go and check her out if you have not seen all of the work they've done with substrates to show the survival of those. We were focused on the mats because we do a lot of microgreen work. And when we looked at mats, and the survival of, in this case, we did basil variety. We did a variety study with basil. What we found is that when we talk about E. coli survival, and if you were to have E. coli in the seeds, so we inoculated the actual seeds, over time, E. coli goes down over time in there. We did this also with salmonella, and we found a very similar pattern of the bacteria surviving both in the roots itself, in the plant itself, and on the mat. So in the microgreen, the growing mat was a great median. In this case, to have the similar pattern of both salmonella and E. coli to be able to survive. So this also brings in the great point of thinking about what your growing medium you are using when you're doing your germination to make sure that that is pathogen free. So let's dive into the nutrient film technique and looking at lettuce. So this is some work that we just completed last fall, where we looked at, in this case, we looked at EC levels to see if there's a difference if how you manage your EC electroconductivity levels within your system. Would that affect the survival of E. coli and salmonella within your system? I can tell you the big point out of that one was EC didn't matter. E. coli, salmonella did not care what the EC was. They still were able to survive. But we did find through this work a significant difference on how E. coli and salmonella behaved in the system. So this is a big graph, a lot of different colors and different codes, but I'm going to break it down a little bit more in the next slides. So in this slide, pink is our E. coli and blue is our salmonella. The blue solid lines are our roots, and the jotted lines, our jotted lines are the roots and then our nutrient film. So let me go a little bit deeper so that you can see it a little bit easier as we go forward. So this is salmonella. This is lettuce again, and this is over 28 days of growing. We inoculated into the water at the very beginning at 0 hour, about 5 logs. What we see with salmonella is it's significantly reduced by day 2. By day 2, it came to undetectable levels using regular detection techniques. But it was able to persist in the system all the way through harvest. So this brings in a great point of making sure whatever your detection method is, that it is sensitive enough. So if your detection method does not detect below 25 cells, then you would say after day 2, there's no salmonella in your system. Whereas there actually is salmonella in your system, and it just was surviving at very low levels throughout a harvest time period. And this picture also shows that same thing, that by day 7, when we looked at the roots itself, it went to undetectable levels as well. So the first one was nutrient solution. This one is looking at roots. By day 7, if you were to test those roots and you didn't have a sensitive test that could detect lower than 25 cells, you would say it was negative, but Salmonella is still thriving in the system. When we look at E. coli, we saw E. coli does a bounce back. So I wanted to go into the mechanism of E. coli because it has an incredible ability to use DNA repair so that it would go down after about 2 or 3 days to undetectable limits, basically recover itself over about a week, and then bounce back into detectable limits. So we've seen this pattern over and over again within whether we're looking at lettuce, basil, other herbs, where it's able to go down to undetectable levels and bounce back. And this is a nutrient solution. In the roots, we see this kind of null. It goes from the solution, it attaches to the roots because it loves the roots, and then it takes a little bit of time, about 7 days, for it to get its bearings and repair itself, and then it bounces back through the conclusion of the cycle. So we see a similar bounce back pattern of E. coli. So really the takeaways from this initial research is Salmonella, it loves to decline quite rapidly to unquantifiable levels, so detection does matter. E. coli, on the other hand, will go to undetectable levels, but we'll see it bounce back. And the roots and the agricultural water were good places to be able to sample. So at this time, I'm going to turn it over to Grace, who is going to come on, and she's going to talk a little bit about deep water culture versus nutrient film technology and some of her research.
Thank you so much, Dr. Walla. So my name is Grace Akumu. I'm a PhD student in Dr. Walla's lab. And part of my research is looking at the survival of Salmonella and Listeria monocytogenes in CEA, specifically deep water culture and nutrient film technique, and then looking at the some interventions. But today I'll be just presenting the survival of these two pathogens in deep water culture and nutrient film technique. Okay, so before I go to the results, deep water culture, most of you must know this, but I'll reiterate for those that are not familiar. In deep water culture, you have the plants sitting on a raft or a board, and the roots are submerged within the stagnant reservoir that has the nutrient solution. And then for the nutrient film technique, you have a thin layer of nutrient solution continuously passing over the roots of the plant, as you can see on the picture on the right. All right, so let's start with the results. So for this study, we introduced Listeria monocytogenes into the nutrient solution after transplanting into deep water culture and nutrient film technique. I'll just be saying DWC and NFT. So we started with a scenario that emulates moderate to high contamination, that is 5 to 6 logs of pathogens. So on this graph, it shows how they survived in nutrient solution, and on the y-axis you have the pathogen concentration, log CFU, and on the x-axis you have time post-inoculation. So we monitored this over 28 days while growing basil. So, Dr. Continue? Yes. So the blue is the NFT and the black is the deep water culture. So from this, we see that the pathogen persistence or Listeria persistence was higher throughout the monitoring period in deep water culture than NFT. And this could be attributed to some of the environmental conditions in deep water culture. Secondly, could you go back a little? Yes. Secondly, we see that in both the deep water culture and the NFT, the pathogens decreased over time. So when they decreased over time, we will see that next. We'll see that yes, in the nutrient solution, it declined to below quantifiable levels by day 12. So here we see the very first line is the limit of quantification, which for most conventional methods is 25 CFU. So by day 12, it falls below quantifiable levels. That means if you're using a conventional method that detects 25 CFU, you will not be able to quantify that. But we went ahead and enriched, and then we were able to detect, we were able to find levels that were there throughout till harvest, which is 28 days. So we see that for both systems, deep water and NFT, they remain detectable till harvest. So we will talk about the implications of this later. The second graph talks about the roots. So we did not inoculate the roots. We inoculated the nutrient solution. Next. So you will see that Listeria persisted longer in deep water culture than NFT, and then it persisted longer in the root zone. So within 24 hours, you see that we see that right here. Yes, while we did not inoculate the nutrient solution, the levels of the pathogen in the roots zone had spiked up to the levels that were in the nutrient solution. That means that the roots act as a reservoir and attract the pathogens. And we will explain why that is the case. Because the pathogens require a carbon sink, require organic nutrients. They require an environment where they can attach and colonize. And the roots provide a favorable environment for that. So here we see the same thing. So in NFT, we see that the Listeria dropped below quantifiable levels by day 24. So that means it persisted longer than it persisted in the nutrient solution. In the deep water culture, we see that Listeria remained quantifiable till harvest. What this means is, in case you have an initial contamination in these two systems, while you may be unable to quantify in the nutrient solution, the roots will remain, Listeria will remain on the roots, and you can still detect it with quantify it and detect it using conventional methods, because it only declined slightly. And for Listeria, 3 logs CFU is still a very high concentration for pathogens. So then we went ahead to look at Salmonella typhimurium. As Dr. Walla talked about at the beginning of the presentation, it's one of the pathogens that has been implicated in so many of the outbreaks in CEA. So we did the same thing. We introduced the bacteria, cocktail Salmonella typhimurium, into deep water culture and NFT. And what we saw was similar in the nutrient solution. However, the persistence was slightly lower than what we saw with Listeria. So here we see that over time, over the 28 days, Salmonella reduced but persisted more in the deep water culture as opposed to the nutrient film technique. So as we can see right here, we also see that in the nutrient film technique, Salmonella declined below quantifiable levels by day 6, which means that your conventional methods would not be able to quantify that. But it was detectable throughout until harvest. In the deep water culture, this only happened at day 24. That means if you're someone who is harvesting, because we were using the Nufar variety. So if you're someone who is harvesting before 24 days, that means you'll still be having quantifiable levels by that point. Then we went ahead to look at the roots. So we see a similar trend that within 24 days, even if we did not inoculate the roots themselves, the pathogens had colonized the roots. So we see that by 24 hours, that is day 1, as we can see here, the pathogens had reached the levels that were in the nutrient solution. Secondly, we see that the deep water culture, that is the black line, we had Salmonella persisted more in the deep water culture than in the NFT. So in NFT, the Salmonella dropped below quantifiable levels by day 24. And in DWC, it remained quantifiable till harvest. So what this would mean is if you still have those pathogens in the roots, in the nutrient solution, by the time you're harvesting, the possibility of cross contamination is there. And then we will talk about other implications on our takeaways. Here we then went to look at what is the implication, slightly behind. Yes. What are the implications of this? How are the pathogens behaving on the roots? Do we have any translocation or internalization? So when we tested this, we found that there was no detectable presence of both pathogens on the leaves. And this was because of the way we were handling the system, because we were handling it aseptically, not to cause cross-contamination over, which is usually not the case with how meticulous we were doing things. So we did not find any detectable presence, because we did that because we wanted to find whether they are actually internalizing and they would move to the shoots. So this means that while contamination is significant in the nutrient solution and in the roots, it does not necessarily transfer to the plant aerial parts. The only way that could happen is a case of cross-contamination, either from the nutrient solution or from the roots, from the handlers or the machines that we're using and the surfaces that we are using during harvest. So from this, we see that the root zone and the nutrients reservoirs were the critical contamination hotspots rather than the edible foliage. So we went ahead to ask ourselves, does the pathogen matter? Like, does the pathogen you have in the system matter? And yes, the findings we had were, yes, it does. When we look at the roots in NFT, we see that we had significant difference in survival of Listeria and Salmonella at 3 points, that is day 1, day 12, and day 28, where Listeria survived better than Salmonella. But when we look at both, we see it's L mono, which is Listeria monocytogenes, survives better in both systems. But the difference in survival is more pronounced in our NFT than in deep water culture. So the key takeaways from this research, one is in case we have initial contamination, pathogens declined faster in NFT than in the deep water culture. And from this, we know it's because deep water culture provides more conducive conditions for bacterial survival. What is the implication of this for people in the industry? I know that both systems, yes, I know that both systems are currently used, but NFT systems may offer a comparative advantage for food safety, but still will require strict controls. Like I said, the pathogens remained detectable. They were there. They were still in the system, but just not quantifiable using conventional methods. But deep water culture systems may require extra monitoring. Then the other takeaway is the root zone acts as a major reservoir. And we call it a reservoir because when I look at the NFT system, is you're having a continuous flow of this nutrient solution back into the reservoir. So if we have the roots colonized, the nutrients solution, the thin film of nutrient solution will keep picking the pathogens and inoculate the NFTs. Then the implication, worker hygiene. Like I said, we didn't have anything in the leaves. That means that most of the contamination you would have on the leaves will be coming from cross-contamination, most likely. So worker hygiene, cleanliness, and caring about the inputs that come into your system, and also the water source quality. Because we are looking at the main points where pathogens will actually get into your system. Next. Yes. The other thing was pathogen persistence despite reduction. So this poses a residual food safety risk because we've seen that a decline in concentration below quantifiable levels does not guarantee complete elimination. So what are the implications? So one is if you have initial contamination, you're likely to have a persistence if nothing is done in the system till your harvest. Secondly is the potential for cross-contamination is there during harvest and during handling. The third is disposal of waste. We never talk about this, but what happens to the nutrient solution after you've harvested your produce? What happens to the roots? Where do you put them? Do we care about any treatment that goes to them after that? So if you have a pathogen like Listeria in your system, and then the roots, we've talked about them being a hotspot, and after you've harvested, you just dispose them. That means you're introducing into the environment this pathogen that's very good at forming biofilms, very good at persisting in environments, whether it's your facility or the environment surrounding your facility. The other thing is bacterial levels are falling below quantifiable levels. They pose a challenge, especially because some of the methods that are currently being used in the industry for detection will not be able to detect one CFU, like one CFU per ml. Yes. So what we need to do is if we have, I know that some of the industry players are coming up with methods that have significantly low detection limits, but as it stands now, it isn't the case. Then the other is food safety interventions should prioritize root level and water system controls to reduce foodborne pathogen risks in hydroponic produce. The other takeaway is Listeria mono is generally more persistent in hydroponics than Salmonella. So for the industry, a one-size-fits-all approach cannot work. So interventions and our protocols, sanitation protocols or schedules and system modifications should work with the worst case scenario, worst case pathogens in mind. Then we saw that the early production phase is the most vulnerable window. So the way your employees or the workers in hydroponics handle produce and handle systems during the first 6 to 12 days of the produce is the most important because that's where we have a very high possibility of cross-contamination. Then if the foodborne pathogen is present, it will persist through the system. So testing methods matter, and I'll let Dr. Walla complete this section.
So I want to
Thank
thank
you.
Grace for describing her some of her dissertation work. We should say that we did reduce this. We were going to talk about living plants as well. We've done a lot of research with the roots attached and the survival. Our take home is it survives through shelf life. But stay tuned. If you're at Cultivate this year, you'll hear a lot about living plants in that work. I want to thank our whole Walla group, as well as my previous collaborators that are at Iowa State, several students, Professor Chris Curry out there, and then Michael Evans. He was at University of Arkansas. We did a lot of this work, and this is USDA funded work that we represented. So with that, we welcome any questions from the audience about our presentation.
Well, thank you so much. Such a useful and science-based information. I really appreciate.